The science of epigenetics has transformed our understanding of aging, revealing that how long and how well we live is influenced not just by the genes we inherit, but by how those genes are regulated throughout life. Unlike genetic mutations, which alter the DNA sequence itself, epigenetic changes modify the activity of genes without rewriting the underlying code. This dynamic layer of regulation, shaped by both intrinsic processes and external exposures, plays a central role in determining biological age, the functional state of our tissues and systems, which often diverges significantly from chronological age. Among the key epigenetic mechanisms, DNA methylation, non-coding RNAs, and chromatin remodeling are now recognized as pivotal modulators of aging, offering not only markers of biological wear, but also potential targets for intervention. DNA methylation, the addition of methyl groups to cytosine bases in DNA, is perhaps the most studied epigenetic hallmark of aging. As individuals age, their methylation patterns become increasingly disorganized. Regions that should remain silenced may become active, while essential genes may be inappropriately suppressed. This dysregulation contributes to inflammation, genomic instability, and loss of cellular identity, all hallmarks of aging. Researchers have developed epigenetic clocks based on specific DNA methylation signatures that can accurately estimate biological age. Notably, studies show that centenarians and their offspring tend to exhibit slower progression on these clocks compared to age-matched controls, suggesting a biological delay in epigenetic aging that may underpin their remarkable health span. The role of non-coding RNAs, particularly microRNAs, miRNAs, and long non-coding RNAs, LNCRNAs, is increasingly appreciated in the orchestration of age-related gene expression. These RNA molecules do not encode proteins, but instead fine-tune gene activity by degrading messenger RNAs or blocking their translation. With age, the expression profiles of many miRNAs shift in ways that promote inflammation, impair stem cell function, and reduce cellular resilience. Yet in long-lived individuals, these changes appear attenuated or selectively adaptive, with miRNAs that suppress pro-aging pathways remaining more active. This suggests that non-coding RNAs help maintain cellular balance and prevent the epigenetic drift that accelerates aging in the general population. Chromatin remodeling, which governs how tightly DNA is wound around histone proteins, further shapes the aging process by controlling access to genetic information. Youthful cells tend to maintain a flexible and responsive chromatin landscape, allowing for robust stress responses and tissue repair. With age, chromatin becomes more rigid and fragmented, leading to reduced transcriptional adaptability and higher vulnerability to environmental stress. However, cells from centenarians and their offspring often retain more youthful chromatin characteristics, preserving the capacity to upregulate protective genes when needed. This chromatin stability may be one reason why these individuals maintain function across multiple organ systems longer than their peers. Importantly, epigenetic aging is not a fixed trajectory. Lifestyle and environmental factors, including nutrition, physical activity, sleep, stress levels, and exposure to toxins, have all been shown to influence the epigenome. Caloric restriction, intermittent fasting, and certain bioactive compounds such as polyphenol, sulforaphane, and omega-3 fatty acids may slow epigenetic drift by modulating methylation enzymes, histone acetylation, and miRNA expression. Likewise, regular physical activity and stress reduction practices like meditation have been linked to more favorable epigenetic profiles, potentially reversing some age-associated changes. Recent clinical studies suggest that it may be possible not only to slow epigenetic aging but even to partially reverse it. Trials using drug combinations, targeted diets, or gene therapies have demonstrated modest reductions in biological age markers, reigniting interest in whether age-related decline can be truly modified at the molecular level. However, these interventions are still in early stages and must be evaluated for long-term safety and reproducibility. Centenarians, and particularly their offspring, offer compelling models for this line of research. Their epigenetic patterns seem to resist the typical trajectory of aging, suggesting a mix of inherited stability and lifestyle-mediated resilience. Studying how their methylation patterns remain intact, how their non-coding RNAs preserve homeostasis, and how their chromatin maintains plasticity could yield actionable insights for the broader population. Epigenetics reframes aging not as an inevitable slide into dysfunction, but as a malleable process subject to influence across the lifespan. It connects the cellular to the behavioral, showing that choices and exposures can leave molecular marks that shape long-term health outcomes. By decoding the epigenetic signatures of longevity and identifying interventions that support optimal gene expression over time, we move closer to extending not only how long we live, but how well we age.